Hello everyone, welcome to today's live broadcast, Photometry Made Easy, Obtaining Reliable Results with Nucleic Acid Measurements. I'm Judy O'Rourke of Lambert, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by Lambert and sponsored by Eppendorf. Eppendorf is a leading life science company that develops and sells instruments, consumables, and services for liquid sample and cell handling in laboratories worldwide. For more information, please visit eppendorf.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or you can submit your problem through the green Q&A button, lower left. I would now like to introduce today's speaker, Natascha Weiss. Natascha Weiss is a field application specialist responsible for the photometry product portfolio within customer support at Eppendorf AG headquarters in Hamburg, Germany. She joined the company in 1999 and worked on electroporators, dispensers, PCR equipment, consumables, as well as several cross-thematic projects. Natascha studied biology at the University of Würzburg in Germany. For her diploma thesis, she employed a variety of molecular biology methods investigating genes coding for virulence factors in the genome of pathogenic bacteria. I will now turn it over to Natascha for her presentation.
Thank you very much, Judy, for your kind introduction. Here's Natascha. Welcome to everyone, and thank you for joining our webinar, Photometry Made Easy. My presentation today will focus on the topics photometry basics and nucleic acid quantification. Regarding the latter, I will cover the introduction to the method, evaluation, and troubleshooting, as well as an overview on the workflow, together with a summary. First, we start with photometric basics now. Which wavelengths are used in photometry? Photometry bases on light, which is a kind of electromagnetic wave. Data in literature vary slightly, but light having wavelengths from 100 to 400 nanometer is called ultraviolet. The range from 400 to 700 nanometer covers the visible light, and above 700 nanometer to near 1 millimeter we find infrared light. As the name suggests, only visible light can be seen as a color with our eyes. White light, like the sun, includes ultraviolet, visible, and infrared light. Such white light may be split into narrow bands of wavelength using tools like a filter or prism. This results in the so-called monochromatic light, and photometric measurements for most biochemical methods are applied at wavelengths from approximately 200 up to 900 nanometer. Different materials absorb light at specific wavelengths. This can be shown in a photometric scan across a large wavelength range, which is shown here by an example of the dye brilliant blue. Spectrophotometry is a scientific method which bases on the absorbance of light by a substance. Thereby, the underlying physical laws are used for estimating the concentration of the substance. For this approach, a peak wavelength, which means wavelength which shows maximum absorbance, is chosen. Most important parts of a photometer are a light source, a place for positioning the sample to be studied, and a detector for determining the light intensity which is transmitted by the sample. An additional element, which is not included on this slide, is a part able to split white light into monochromatic light. In a photometer, incident light with the intensity I0 passes through a sample. The light which is not absorbed by the sample is transmitted and measured by the detector. The intensity of transmitted light is termed I. The ratio of I to I0 gives the transmittance, which is often expressed as a percentage, resulting in the percentage transmittance. The software of the photometer can convert the amount of light that is transmitted into the amount of light that was absorbed by the solution. The underlying formula states that the absorbance corresponds to the negative decadic logarithm of the transmittance. In a photometer, the detector determines the absorbance by processing the information of incident light intensity and measured transmitted light intensity. Absorbance, which is measured, depends on three parameters. C stands for the sample concentration and L for the optical path length, which is defined by the cuvette used. It describes the distance traveled by the light. Epsilon is the molar extinction coefficient, which is a physical constant of a molecule. It is used to define its ability to absorb light of a specific wavelength. I will come back to this coefficient later. The Lambert-Beer Law describes these correlations and represents thereby the basis for photometry. It is a combination of the Lambert's law, which states that the absorbance of a sample is directly proportional to the distance traveled by the light, and the Beer's law, which says that absorbance is proportional to the concentration of the sample. State-of-the-art photometer includes software programs based on the reformed Lambert-Beer Law, which enable the direct calculation of the concentration of specific sample types. Why measure absorbance instead of transmittance? The aim of spectrophotometry in biochemical applications is to determine the concentration of substances. So why do we prefer to express the Lambert-Beer Law using absorbance as a measure rather than percentage transmittance? On this slide you see a series of samples, each of them absorb 50% of the incoming light. So the amount of the original radiation passed by the first sample is 50%. By the second sample is 25%, and by the third sample is 12.5%. By plotting the transmittance as well as the absorbance, we see that percentage transmission leads to an exponentially decaying curve, whereas the absorbance values, which base on a logarithmic formula, result in a linear graph. We have seen that the amount of light diminishes exponentially as it travels through the material. Therefore, transmittance is obviously difficult for determination of sample concentration. Since the absorbance of a sample is defined by a logarithmic formula, it is directly proportional to the concentration of the absorbing material. This linear relationship enables an easy and convenient quantification of samples. In order to get a feeling for the meaning of absorbance units, I put common photometric values with corresponding percentage transmittance rates together on this slide. You see that due to the logarithmic formula, zero absorbance originates from a transmittance rate of 100%. An absorbance of 1 is equal to 10% light transmittance, and an absorbance of 2 corresponds to 1% transmission. Much higher absorbance values will not be suitable, since any further increase in concentration will reflect only slight changes in absorbance, and the amount of light which reaches the detector will be too low. So this leads to the question, why the absorbance range is reasonable in photometry. There is a linear dependency between absorbance and concentration, but this linearity is limited by chemical and instrumental factors. The Lambert-Beer Law is not valid for high concentrated samples due to strong molecular interactions, and limitations are given by stray light effects of the photometer used. Stray light can be caused by scatter or reflection of light inside the device. When reaching the detector, it will severely impact high absorbance measurements. In the left figure, the influence of different amounts of stray light is shown by a plot of measured absorbance values versus true absorbance. Only when no stray light is available, which is, of course, theoretical, total linearity can be achieved. The curves of measured absorbance will start flattening the earlier, the higher the stray light component of the photometer. This means that the concentration of high absorbing samples will be underestimated. According to this example, readings up to 1 are perfectly linear. Devices with a low stray light rate may also give reliable values up to 2 or 3 absorbance units. Besides deviations when measuring high absorbance, the Lambert-Beer Law is also limited when very low concentrated samples are used, due to background noise coming from electronic components of the photometer and the environment. The figure on the right side illustrates that an absorbance higher than 0.1 can be easily distinguished from the noise, whereas this becomes more difficult with lower sample concentrations. It has to be considered that the percentage of the error increases with low concentrations, and you have to decide by yourself which error is acceptable for your application. For being on the safe side, your values should lie between 0.1 and 1.0 absorbance units. All substances, if clear or not, absorb some light. Therefore, we have to consider that light is also absorbed by the cuvette and the sample solvent, if we want to determine the concentration of a substance. In order to obtain the absorbance of the sample only, a blank measurement has to be performed using the same wavelength as well as the same cuvette and solvent prior to sample measurement. The blank measurement sets the instrument to 0, which corresponds to a subtraction of blank from sample, resulting in the true sample value. Before we start with the next chapter now, I would like to ask you to participate in our first poll, which you will see soon on the screen. So you see the result of your poll, and we see that more than 72% of you are already doing nucleic acid quantification, and additionally 14% plan to do it. I would like to go on with the next part now, nucleic acid quantification. And I will introduce the method first. Why quantitate nucleic acids? Nucleic acids are isolated in order to use them for a high variety of downstream applications. In general, these reactions require a specific amount and a high quality for optimum performance. Therefore, it is crucial to evaluate the concentration of the nucleic acids as well as their purity after isolation. Especially the presence of inhibiting contaminants, which may come from poor sample preparation or which are associated with a difficult sample matrix like blood or soil, can lead to visible problems. Extra products may be generated during PCR analysis. Activity of enzymes can be reduced. Transfection shows low efficiency, or short wavelengths during sequencing can occur. An easy method to perform quantification and quality control of DNA and RNA is represented by UV-Vis spectrophotometry. Nucleic acids show an absorbance peak at 260 nanometer, which allows for a simple estimation of the concentration. After absorbance measurement, the software of the photometer can directly calculate the concentration of the sample basing on the Lambert-Beer Law. This example shows a measuring result of double-stranded DNA for which the sample-specific factor 50 microgram per milliliter is used. On this slide, I would like to give you some more information about parameters of the Lambert-Beer Law. The molar extinction coefficient epsilon is valid for specific sample types at a specific wavelength. For calculating concentrations easily, the reformatted Lambert-Beer Law is used, and the reciprocal of epsilon combined with the path length is replaced by the factor f. The underlying statement says that an absorbance of 1 corresponds to a concentration of 50 microgram per milliliter double-stranded DNA in a 10 millimeter cuvette. In the table, the factors respective to the concentration at an absorbance of 1 are indicated for different nucleic acid types. The factor is not only influenced by the sample type, but also by the path length of the cuvette. When smaller cuvettes with 1 or 2 millimeter path length are used, the factor will increase. Examples for different cuvette types are given in the second table. It is shown that smaller path lengths are suitable for the detection of higher concentrated sample solutions. A number of other substances can interfere with measurements of DNA or RNA since they also absorb light at 260 nanometer. This leads to an artificial increased result. To compensate for this, purity ratios and background correction have been developed to help eliminate false readings. Potential contaminants like proteins, carbohydrates, salts, and organic solvents show high absorbance at 230 nanometer and 280 nanometer. These wavelengths are used to calculate the purity ratios A260 to A280 and A260 to A230. For determining the background, a measurement at 320 nanometer or 340 nanometer is typical, since nucleic acids do not absorb in this range. On this slide, some more information on most important contaminants is given. Proteins are probable contaminants after isolation of nucleic acids. They have an absorbance peak at 280 nanometer, and additionally absorbance at 230 nanometer is very high. The presence of other impurities like carbohydrates, catrabic salts, and organic solvents is indicated by high absorbance at 230 nanometer only. A mixture of different nucleic acid types like DNA and RNA cannot be distinguished in absorbance measurements since they show the same peak at 260 nanometers. This can be crucial when, for example, plasmid DNA is isolated from bacteria without RNase digestion, since 90% of the purified nucleic acids are RNA. The resulting calculated DNA concentration based on absorbance measurements will lead to wrong results due to massive overestimation. Since RNA shows higher absorbance than DNA, the real amount of DNA will be even lower than indicated. We have seen on the slides before that contaminants absorbing at 260 nanometer lead to vastly increased readings too, and regarding the absorbance range, we have learned that the evaluation of nucleic acid concentrations lower than the microgram per milliliter range is not appropriate. These problems can be overcome by an alternative quantitation method, the fluorescence spectroscopy. In fluorescence spectroscopy, the fluorescence intensity of a dye that binds to a nucleic acid is measured. Incorporated dyes absorb light of their specific excitation wavelengths and will afterwards emit light of a lower emission wavelength. Fluorescence dyes can react very specifically. The dye pico-green, for example, only binds to double-stranded DNA and not to other nucleic acid types or contaminants. Upon binding, the fluorescence of pico-green increases more than 1000-fold, which leads to a high sensitivity of the method. Taking these facts together, a high accuracy in nucleic acid quantitation can be achieved. Evaluation of such an experiment is not done by a factor, but using a standard curve. Therefore, nucleic acid standards with a known concentration as well as the samples are labeled with a fluorescence dye. The standards are measured in order to generate a standard curve. Afterwards, the concentration of an unknown sample is calculated by comparing the intensity to the emitted light with the standard curve. If we compare the methods, we see that both have their advantages and disadvantages. So the decision which one is most suitable will depend on the sample type and the applicational requirements. The detection limit for fluorescence measurements is quite low, so it enables the quantification of highly diluted samples. Additionally, the method is very specific regarding the target molecule and therefore suitable for mixed sample types. Information on sample purity and possible interfering substances are only available with absorbance measurements, which are fast and easy to perform and which do not require expensive reagents. For sensitive downstream applications, it may be useful to perform both types of measurements in order to achieve a very accurate quantification in combination with information on the quality of the sample. We come now to the next poll. It refers to all who already do nucleic acid quantification by photometry. So the result of this poll is here, and we see that 65% of you use only UV absorbance, and 3.4% already do fluorescence measurement, whereas 31% perform, of course, the two measurement methods. On the following slides, I would like to present how to analyze measuring results and in which way troubleshooting is done. Therefore, we will follow this evaluation sequence by answering the questions: which measuring range is readable, which are the causes of background, in which way purity ratios are evaluated, as well as how to make sure that repeated readings will be stable. In the part Photometry Basics, we had already seen the slide which shows that very high absorbance as well as very low absorbance is error-prone. The linear range of measurements depends also on the technical specifications of the photometer used, but in general, valid data can be achieved when the readings are between 0.1 and 1. Absorbance readings, which are outside the linear range, are not directly proportional to the sample concentration, and this leads to inaccurate results. High absorbance values result in stray light effects in the photometer. In order to reduce the absorbance, samples can be either diluted or a cuvette with a shorter path length can be chosen. Low absorbance values are influenced by background noise. For achieving better accuracy, it is recommended to concentrate the sample or to increase the cuvette path length as possible. If only low amounts of low concentrated sample are available, also evaluation by fluorescence can be an alternative. Background is the turbidity of the sample, and it is measured at a point of the spectrum which is not related to the sample absorbance. For UV spectrophotometry of nucleic acids, typical background wavelengths are 320 or 340 nanometer, since nucleic acids will not absorb in this range. So the value should be close to 0. Background may be due to light scatter caused by contaminant particles, air bubbles, or by precipitates in the sample. The same effect comes from a dirty cuvette or cuvette shaft or a low liquid level in the cuvette, which can lead to a meniscus in the height of the light beam. Absorbance at 320 nanometer indicates a contamination and is a hint that the measured concentration is not correct. Background values pretend a higher concentration of the sample and falsify purity ratios. Additionally, contaminations like microbial growth can affect downstream reactions negatively. For relatively low background readings, you can just activate the background correction. This will correct all relevant data by simple subtraction. If higher values than approximately 0.03 are seen, you should have a closer look on your sample in order to find the reason for light scatter. Often, a simple centrifugation step, removing air bubbles or the resuspension of sample precipitates is sufficient. But sometimes, further purification of the sample may be necessary in order to avoid an influence on downstream applications. Now we come to the purity ratios. In advance, I would like to say that different values are found in the literature regarding purity ratios of pure nucleic acid samples. One explanation for this fact is that samples are used which are resolved in different buffers, and the pH value of the buffer has an influence on the ratio. On the other hand, ratios from literature are ideal ones, and there are diverse samples like body fluids or soil samples which will never meet these values. So the ratios are helpful for determining the quality, but they will not replace your own rating on sample quality resulting from your experience. The most common purity check for DNA and RNA is the ratio of measurements at 260 and 280 nanometer, since proteins have a maximum absorbance at 280 nanometer. For DNA, a ratio of 1.8 to 1.9 indicates a good level of purity, whereas an RNA sample is considered pure for a ratio at 1.9 to 2.0. This is due to its higher ratio of uracil in RNA as compared to thymine, which is a component in DNA. Lower values indicate impurities from proteins, as well as from aromatic compounds like phenol, which shows an absorbance peak at 270 nanometer. A number of substances absorb at 230 nanometer, as this is the region of absorbance of peptide bonds, aromatic side chains, carbohydrates, chaotropic salts, or organic solvents. Values between 2 and 2.5 for the ratio 260 to 230 are typical for a pure sample. Besides solid contaminants, different solubles may be found in sample solutions too. Sources are the sample matrix with proteins as main contaminant, but there are also carbohydrates from plant material or humic acids from soil samples. In addition, traces of reagents used during the isolation of nucleic acids are found. Typical examples are phenol and salts like GTC. The purity ratios are used as an indicator in order to decide if the isolated nucleic acids are pure enough to be used in downstream applications. Especially organic solvents and high salt concentrations can show inhibitory effects which lower the efficiency of genotyping experiments or the transfection of sensitive cells. Also important is the fact that these contaminants absorb at 260 nanometer too and will lead to an overestimation of nucleic acid concentration. Typical contaminants are proteins on the one hand and salts like GTC, which are used for the isolation process. As seen by the DNA spectra and in the table, a typical indicator for protein contamination is a lower ratio 260 to 280, together with a strong reduction of 260 to 230 values. When only 260 to 230 values are low, contaminants like GTC are the cause. I have mentioned before that blank measurements are necessary to subtract the absorbance from cuvette and solvent from sample measurement. Therefore, it is important to use exactly the same solvent for blank as well as for sample. The images on this slide show what happens to the DNA spectra when different solvents for blank and sample have been used. In the first example, water was taken as blank solution and tris buffer was the sample solvent. In the second example, the solutions were used vice versa. We see a high impact on the spectra, which are far away from a normal DNA absorbance spectrum, as well as on the purity ratios 260 to 230. In the first example, additionally, the ratio 260-280 is slightly increased, whereas the second example shows a sample concentration which will be more than twice as high as the real value. From this result you see that it is extremely important to use the same solution for blank as for the sample solvent. On this slide, I have summarized different purity ratio combinations with typical reasons leading to this effect, together with recommendations how to handle them. If both ratios are low, probably contaminations with proteins or traces of aromatic compounds like phenol are the reason. If required, the nucleic acid can be purified, for example by phenol chloroform extraction, if proteins are available, or by chloroform in order to remove phenol. Residues of salts, organic solvents, and carbohydrates have only an effect on the ratio A260 to A230, which is reduced. Also, these components can be removed by further purification. In case of excessive salt content, for example, an alcohol precipitation is appropriate. Different effects are seen if the blank and sample solvents are not the same. If the blank consists of tris buffer and DNA was solved in water, a strong increase of the ratio A260 to A230 is seen. The other way around will show a slight increase of 260 to 280, whereas A260 to A230 is too low. In order to achieve reasonable results, same solutions have to be used. If the sample solvent is not known, it may be required to precipitate and resolve the sample. If high amounts of RNA contaminate a DNA sample, only the ratio A260 to A280 is slightly increased, and RNA's digestion will help in this case. Also, fluorescence spectrometry can be a solution in many cases in order to achieve accurate results. Sometimes a single measurement will not reveal any problems at first sight, but when you read a sample multiple times, you see different results. Such fluctuating values can be induced by particles in the air bubbles as well as by a drift of the pH value. However, the most important reason is insufficient mixing after resuspension or dilution steps, which lead to inhomogeneous solutions. The figure demonstrates that mixing the sample carefully is very important in order to receive reliable results. For this experiment, 10 microliter of DNA solution have been added to 90 microliter buffer. Measurements are repeated every 5 seconds. It is obvious that no mixing results in a heterogeneous solution, but even after 3 times dispensing with a pipette, the readings differ a lot. On the other hand, it is shown that vortexing is very effective in order to achieve stable and reliable readings. Besides careful mixing, the cuvette has to be checked for air bubbles, and, if possible, a slightly alkaline buffer should be used as a sample solvent in order to avoid pH shifts. Now we come to the third poll, and it would be great if you can give us your answer about how often you face problems in nucleic acid quantification. So from the results, we see that only 4% never face problems, whereas 60% rarely, and 20% encounter frequently problems. Now I would like to come to the last part of my talk, which covers the workflow and a short summary. When setting up a photometry experiment, it is recommended to use a calibrated pipette with corresponding tips in order to ensure that dilutions are prepared correctly. For blank and sample, the same solvent has to be used. Suitable is a slightly alkaline buffer like tris, since also the pH value can affect sample readings. Avoid dilutions higher than 1 to 50, since they result in a higher error probability. After dilution, carefully mix the sample, if required, in combination with a short centrifugation step. A cuvette which is used for photometric measurements has to be clean and intact. Generally, this is given for disposable cuvettes, but when using quartz cuvettes, take care to clean them carefully and check on a regular basis for scratches. For measuring nucleic acids, UV transparency is of course a prerequisite, but also make sure that the light path height corresponds with the light beam height of the photometer. 4 wavelengths are important for determining the concentration and inform about the purity of the nucleic acid sample. Nevertheless, a complete spectrum can provide additional information regarding potential errors in the handling or contaminants. When programming the photometer, have a look on the following settings: the path length of the cuvette you take, the unit of your results, and the right factor according to your nucleic acid sample. For evaluation of the quality, purity ratios as well as background absorbance are informative. Please ensure that the minimum volume required for the cuvette type is used. It is advisable to take always some microliter in excess in order to avoid a meniscus effect. Not only large air bubbles like the ones shown on the picture, also smaller ones, which are not visible at first glance, can influence the reading. Therefore, check always blank as well as sample solution for air bubbles prior to measurement. After reading the samples, it is important to evaluate our results. First and most important point is whether absorbance values are within the linear range of the photometer. Then look for background at 320 nanometer. Low values up to approximately 0.03 may be corrected. That means they are subtracted from all other results. If the absorbance is higher, it is advisable to look for the cause of background absorbance. If these 2 parameters are fine, have a look on the purity ratios and optionally the spectrum. As I have said before, be aware that the purity ratios from literature are ideal ones and they may not be met in every case. You have to define your own acceptable range depending on the sample matrix and the requirements of your applications. It can also be helpful to check by multiple measurements of the sample that no drift or fluctuating values occur. This is now the last slide of my presentation with a short summary of the present content. Be aware about the requirements of your downstream application regarding the concentration and purity of the nucleic acid sample. This does not only affect the choice of the purification method, but also the decision on the method for quantification. Questions to think about are the expected concentration and potential contaminants which can lead to increased readings or may have an influence on follow-up reactions. For the quantification, the following procedure is reasonable. The setup has to be done carefully with regard to pipetting accuracy and mixing. The appropriate equipment consisting of device and cuvette has to be chosen. Programming of parameters and measuring of blank and samples have to be done in the right way. And last but not least, evaluation of results according to the sequence presented before is helpful. So thank you very much for your attention. We have now a look on your questions and we'll start to answer them in the following session. Questions we cannot cover today will be answered later via email. In the meantime, you may have a look on the last poll where you can rate the content of this webinar. I thank you in advance for your feedback.
Thank you for that informative presentation. Before we get started on the question and answer session, I'd like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. The first question is, what can I do if I don't know in which buffer my sample has been solved?
Yeah, if you have a high concentrated sample, I would generate a dilution with a defined buffer and measure this solution instead. Then the effect of the original buffer may be negligible. Otherwise, trying a buffer which is commonly used in your lab for eluting or resuspending samples may be reasonable. In this case, I would recommend to check the complete spectrum. If it looks fine, you can assume that the right buffer was found. If the results are not satisfying, the best way will of course be to precipitate the sample and resuspend it again.
What is the difference between light path and light path height?
The light path height of a photometer indicates the height at which the light beam of the photometer passes the cuvette. So it is important that the light path height of a photometer and the height of the optical window of a cuvette match to each other. On the other hand, the light path or light path length of cuvettes is a measure for the traveling distance of the light through the sample, and you have to set this parameter in the photometer so that you can get correct measuring results.
How does ethanol affect the shape of the absorbance spectrum of nucleic acids?
Ethanol shows a peak which is near to 270 nanometer. This can lead to a slight shift of the absorbance spectrum towards this peak, but only if there are very high contamination. I haven't observed so far significant effects with only traces of ethanol.
The next person says, it happens sometimes that I see a negative absorbance. What can be the cause for it?
Yes, this can occur when the blank measurement has a higher absorbance than the sample solution. A very common reason for it are a mix-up of blank and sample solution, or a dirty cuvette which is used for the blank. Also, air bubbles in the blank can lead to such negative sample values.
Could you explain again why there are different factors for different types of nucleic acids?
Yes, of course. The absorbance coefficient epsilon is defined by the optical behavior of a substance at a specific wavelength. Nucleic acids have a similar structure, but they are not identical. The base uracil, for example, which we find in RNA, has a different structure than thymine, which is the component in DNA. Also, double-stranded molecules and single-stranded molecules absorb light in a different way. And this is the reason why we have different values for epsilon and resulting different factors for RNA, single-stranded and double-stranded DNA.
Why do you recommend a correction for background values up to 0.03 but not for higher values?
This is because an absorbance up to 0.03 is relatively low, but nonetheless significant. A correction of the measuring wavelength will improve the accuracy of results, the concentration, as well as the purity ratios. This will of course work for higher values too, but there is also a high risk that potential contaminants will affect downstream reactions negatively. And from this reason, it is advisable to look for the cause of the background if higher values occur. This is better than just correcting the values.
Can you get accurate results with absorbance measurements? Hang on one second. Can you get accurate results with absorbance measurements only, or is it better to perform a fluorescence measurement in addition?
I have shown in the presentation that both methods have their advantages and, of course, their disadvantages. If you have a very pure sample with a concentration resulting in values in the linear range of the photometer, you can receive very reliable data with an absorbance measurement. But on the other hand, if the concentration is too low, only fluorescence is useful and can be used. However, in many cases, it can be helpful to perform both types of measurements, especially if you need to know if the sample is pure enough for your downstream reaction and if you need a very accurate determination of the concentration.
I would once again like to thank Natascha Weiss for her presentation. Do you have any final comments for us?
So I would also say thank you again for your attention, for your questions, and as I've said before, we will take care for answering all your questions via email after the webinar. Thanks again.
Thank you. I would also like to thank our sponsor, Eppendorf, for making today's educational webcast possible. And as Natascha said, if we didn't have time to get to your question today, we'll be sure to follow up with you. Today's webcast will be available for on-demand viewing through July of 2016. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.